Dr. Cleves received his BS in biology from the University of Arkansas and Ph.D. in molecular and cell biology from the University of California, Berkeley. For his Ph.D., he studied the molecular and genetic basis of morphological evolution in stickleback fish. After his Ph.D., he continued his postdoctoral work studying coral symbiosis at Stanford University. There, he applied next-generation sequencing and genome editing tools to study the genetic works that orchestrate this symbiosis in corals. These technological advancements allow for the dissection of the coral gene function for the first time. In 2021, Dr. Cleves started his own lab studying the cellular basis of coral symbiosis and bleaching at the Carnegie Institute for Science, Department of Embryology, and Johns Hopkins University, Department of Biology. Welcome, Dr. Cleves, and let's hear his fascinating research on coral symbiosis.
Oh, Dr. Cleves, if you could unmute yourself.
There we go. You can imagine after all this time, we'd get better at that. I thank you so much for inviting me to give the talk, and also thank you for facilitating the rescheduling. I'm really happy to tell you about the work that we've been doing to understand the molecular and cellular basis of cnidarian-dinoflagellate symbiosis and its breakdown. And I'll tell you about some of the work that we're doing in my lab, and also some of the work that I did in my postdoc at Stanford. So if you ever spent time on reefs, you've probably been blown away by the incredible amount of biodiversity that exists on these ecosystems, and the ability of corals to build these three-dimensional structures that you can see from space is largely due to the fact that corals have a really crazy endosymbiotic relationship with dinoflagellate algae. And so corals are colonial animals that have a mouth and a gastric cavity. They have two cell layers. They have a gastroderm and an epiderm. And inside the gastrodermal cells, they have resident algal symbionts, so eukaryotes that live inside of these cells. And now these symbionts actually are critical for nutritional aspect of coral reef ecosystems and provide nutrition to the corals. So algae are hit by light, undergo photosynthesis and fix carbon. They translocate carbon from host-derived vesicles called the symbiosome across these membranes to feed the coral, glucose and sterols, for example. And in return, the algae get inorganic nutrients from the host. And this is an absolutely critical symbiosis for corals. And this nutritional exchange lets them to live in nutrient-poor tropical waters. And we know that it's a critical symbiosis in part due to the fact that the symbiosis breaks down upon thermal stress. So when in a process called bleaching. So when water temperatures get 1 or 2 degrees higher than what a coral typically sees in a region, they'll start to expel their algal symbionts from the gastrodermal cells. And if the water temperatures stay high and the corals aren't able to repopulate their tissues, they'll starve to death and die. Now, mass bleaching caused by human climate change has really accelerated the frequency and duration of these bleaching events worldwide. And we've seen massive collapse of our ecosystems, coral reef ecosystems. For example, in 2016, we lost about 30% of the Great Barrier Reef due to an abnormal heat wave that lasted a few weeks. And just to put that into perspective, the Great Barrier Reef is about the size of Italy. So losing about 30% of the Great Barrier Reef in about 2 weeks is just a tragedy, and also a sign of the importance of the symbiosis. Now, despite the effects that climate change is having on our ecosystems, we know very little about the molecular and cellular events that orchestrate the symbiosis. We don't know anything really about the genes that cause the breakdown of symbiosis due to heat stress and bleaching. And the reason we know so little, despite extensive efforts to study the process, is that corals are notoriously difficult to study in the lab. So I usually make a joke about how, trying to take a coral like this into the lab and look at it under the microscope is technically very difficult. There's other aspects of their biology that make them kind of hard to study. For example, they come in inconvenient sizes, they're difficult to grow, they have these hard calcareous skeletons that makes biochemistry and imaging difficult. Bleaching causes death, so trying to figure out what causes symbiosis and the breakdown of symbiosis is very difficult because the animals just die. And then there's only seasonal access to larvae. So the second half of the talk, I'll tell you about trying to build reverse genetic tools in these organisms. We only have seasonal access to larvae for genetic manipulation. So in my lab, we've been mostly focusing on a laboratory model system to study aspects of coral symbiosis and bleaching. We chose the small anemone Aiptasia as an excellent candidate model for these processes because we think that Aiptasia have a lot of aspects of their biology that make them really attractive for a laboratory system. For example, they have the exact same types of symbiosis that are found in corals. We can actually take algae from corals and infect our anemones with them, and they'll form a stable symbiosis, showing the things that we learn in corals are probably, or things we learn in Aiptasia are probably true in corals. We can grow them rapidly in convenient size ranges, and they can fit in 96-well plates. They're soft-bodied. We can grow them with or without their symbionts, so in the symbiotic or aposymbiotic state. And we also induce them to spawn in the lab year-round. So we have spawning every week in my lab. And we can also recapitulate these bleaching events that we're seeing on the world's reefs in a petri dish with Aiptasia. So we could take Aiptasia and we can move them from their normal 27 degrees to 34-degree heat stress, and you can watch bleaching over time. And so here's an anemone that's experiencing heat stress, and as you can see, they gradually lose their algal symbionts, and we're monitoring algal symbionts here by the loss of red chlorophyll fluorescence. We can also use more sophisticated measurements to quantify bleaching. For example, in this plot, we're showing bleaching of Aiptasia upon heat stress at 34 degrees using flow cytometry. And so when I first got into this field, I was really interested, and we're still really interested in trying to understand what genes are involved in the bleaching process. And so we did a really simple-minded experiment where we did an RNA-seq time course of symbiotic and aposymbiotic anemones during heat stress. And so we thought this experiment would allow us to find gene expression differences that are responsive to heat that are dependent on the presence of the symbiont. So the aposymbiotic anemones, again, don't have symbionts. We got a lot of data from these types of time courses, and I'm going to tell you two brief stories. First involves the early heat stress response, and the second is something I call a story about symbiosis-associated genes. I'll first start with the early heat stress response. So when I subjected symbiotic anemones to heat stress, I found that there was a pretty massive and dramatic shift in gene expression in the first 3 hours of heat stress. And so what you're looking at here is a heat map of gene expression in the symbiotic time course. Time under heat is on the x-axis. And you can see that at the first 3 hours, you have an extensive upregulation of genes. And these genes cluster into two types of genes. So genes that come up and then come down very quickly, and then genes that come up and stay up. And so because of this differences in expression between these two heat responsive genes, I was curious about what types of genes are in these profiles. And so I did GO-term analysis and found that cluster 1, the genes that come up and then go down quickly, are enriched for genes involved in apoptosis and the immune system. And cluster 2 turned out to include genes that are kind of the who's who of the heat stress response, including genes involved in protein folding and the unfolded protein response. And so here are some examples. Again, it's kind of the classic traditional examples of genes that are responding to heat in other organisms, including caspases, hypoxia stress genes, heat stress response, and immune-related genes. And so then we compared these heat stress responsive genes to our aposymbiotic time course. So these are the animals that do not have symbionts. And we found a really striking result, which is the majority of these genes are rapidly also upregulated in response to heat stress in aposymbiotic anemones. And this is important because this suggests that this heat stress response is due to the core machinery of the animal rather than to the presence of the symbiont. And here's some more of the illustration of the types of genes that go up and stay up in response to heat stress. We were able to find some differences. So if we looked really hard, we could find some genes that seem to go up in symbiotic anemones, but not aposymbiotic anemones, and some genes that go up and stay up in cluster 2, that in symbiotic anemones, but not aposymbiotic anemones. However, we found 0 genes that are differentially expressed between apo and sim animals that were obviously related to the reactive oxygen stress response. And that's really important because the current dogma of coral bleaching in the field has to do with ROS produced from the algae triggering bleaching. And we saw no evidence of that in our system, suggesting that other mechanisms of bleaching need to be further explored. And this is consistent with other studies that came from the lab showing that bleaching can happen in the light and in the dark. So photosynthesis of the algae doesn't appear to be required. And so it really draws into question this idea that ROS is the major trigger in all bleaching cases. when I was looking at these plots, I was kind of struck by how dramatically and tightly regulated these genes are. So I hypothesized that maybe some of these genes are co-regulated by single transcription factors. And so I wanted to ask whether or not there's a master transcriptional regulator of this early heat stress response. And so what I did was I took I did promoter analysis to find putative transcription factor binding sites. So I took the top 100 heat stress responsive genes in Aiptasia. I looked 500 base pairs into their putative promoters and screened for motifs that might be indicative of a transcription factor binding site. And I found two such sites. One is a nearly completely perfect match to the NF-kappa B protein. NF-kappa B is a master regulator of the immune system. And the other hit is, shouldn't be surprising, this is a heat shock factor 1, which is a known master regulator of the unfolded protein responsive to chaperones. And so this is really exciting to me because I found two clear master regulators, one that's controlling the immune genes and one that seems to be controlling protein folding genes. Both of these transcription factors are upregulated themselves in response to stress. And indeed, if you rank the genes genome-wide by the presence of these transcription factor binding sites in their promoters, you can see that the genes that have the most NF-kappa B seem to be in cluster 1, which are upregulated very quickly in response to heat stress and then go down. And then genes that have the most HSF1 sites seem to be in cluster 2, which are upregulated quickly but stay up. And so we think what we've been able to find is that there's two transcriptional profiles that are upregulated in both sim and apo animals that are responsive to heat stress, and they're partially independently controlled by NF-kappa B and HSF1. Now, these proteins shouldn't be surprising for being involved in heat and immunity. I mean, HSF1 is called the heat shock transcription factor. But the fact that it's well studied made us ask whether or not we could actually use the fact that it's well studied to ask whether or not it has a role in bleaching. And it turns out that there's a chemical inhibitor of HSF1 binding its targets. And called CRIB11. And so what we did is we took CRIB11, which is an inhibitor of HSF1. We treated with the inhibitor in the first 12 hours of heat stress, and then we looked at bleaching after 36 hours. And here are the results of such experiments. On the y-axis is bleaching, percent bleaching relative to the control. And then we have no temperature stress at 34 degrees with or without the drug. And so what you can see here is that the drug at normal temperatures doesn't cause any bleaching response after 48 hours. But the drug that inhibits HSF1 seems to make bleaching worse. And we interpret this by saying that that CRIB11 is actually blocking HSF1 activity. And now we're following this up with RNA-seq experiments to see which targets of HSF1 are affected by the drugs. And so this leads us to this model that HSF1 and the launch of this protein homeostasis pathway is actually required to maintain the symbionts during heat. We also are curious about the role of NF-kappa B and the role in symbiosis or thermal tolerance. NF-kappa B is a master regulator of the immune system, and it's been hypothesized that the immune system might be involved in regulating this internal symbiosis. This kind of started with a collaboration, Tom Gilmore in Boston University, where we took aposymbiotic larvae. So these are larvae that are normally aposymbiotic, and we stained for NF-kappa B protein here in green. And then we infected these aposymbiotic larvae with symbionts, and we found that there was a massive downregulation of NF-kappa B in response to heat stress. And so it leads us to a really interesting model that maybe the NF-kappa B is actually controlling symbiont state and might have a direct role in the symbiosis. So the next story I'll tell you about in terms of this RNA-seq data set was a set of genes associated with symbiosis. So in previous studies, we've been able to identify a set of genes that are massively different between animals that have a symbiont or not. And here are three of those genes: a glucose transporter, a sterol transporter, and an ammonium transporter, that are all up in symbiotic anemones relative to aposymbiotic anemones. And we hypothesize that these transporters are important for translocating nutrients across the symbiosome membrane. Now I was curious about these genes and others that are differentially expressed between apo and sim, how they behave during heat stress as the anemones bleach. And so how I did that is I just called differentially expressed genes between our apo and sim time courses at time point 0, and then tracked those differentially expressed genes through the time course. And I got a pretty non-surprising result, that as the symbiotic anemones bleach, most of their gene expression profiles converge to the aposymbiotic anemones, because showing that these genes are likely involved involved in the symbiosis because the symbiotic anemones are becoming more like aposymbiotics anemones as they lose their symbionts. But we were struck by the timing of this. And so we found that about at 12 hours of heat stress, many genes were already at the aposymbiotic level. And that was really interesting because the animals really hadn't started bleaching at that point. So they haven't started losing their symbionts. So what we see is a massive downregulation of symbiosis-associated genes kind of before bleaching. And that brings us to this model that maybe the downregulation of these genes are actually causing bleaching themselves. And this is really interesting. If you look at what types of genes behave this way, it turns out our ammonium transporter, our glucose transporter, and our sterol transporter are all downregulated very quickly in response to heat stress. And we think these are important for the nutritional exchange. And it kind of leads us to this model that the downregulation of these symbiosis-associated genes might be triggering bleaching themselves. And so just to give a little summary of this RNA-seq analysis, we found an upregulation of heat stress genes, partially controlled by NF-kappa B and HSF1 that happens at 3 hours. We think that aspects of this early heat stress response is protective given our HSF1 chemical inhibitor experiments. At 12 hours, we see downregulation of symbiosis-associated genes, and that seems to happen before bleaching happens at the cellular level. And we think that the downregulation of these symbiosis genes during heat is actually triggering the bleaching response. And so we think we've been able to, and there might be a relationship between the NF-kappa B response and this breakdown of symbiosis based on the connection between the immune system. So in these RNA-seq experiments, I've been able to present a lot of interesting hypotheses about how the symbiosis might occur. However, how could we ever test the role of any of these genes? And so I started to think about how we could begin to test the hypothesis. And this kind of led me to realize that we really need genetic tools to functionally test candidate genes in these systems. So we need to be able to overexpress genes and knock down genes to see what they do to test these models that are being derived from such experiments with sequencing. And so during my postdoc, I've been able to develop several methods to manipulate Aiptasia. The first method that I developed was a microinjection method. What you're looking at here is a needle that's going into the one-cell zygotes. So we have animals that are spawning every week in the lab, and we can inject reagents into the lab or into the eggs. And we also have electroporation methods going. This has been in collaboration with Chris Rinicky, where we can now introduce transformation reagents into hundreds of zygotes at a time, and it's much less laborious than the microinjection method. So just to give you kind of a flavor of the types of experiments we can now do with these two transformation methods, we can actually overexpress genes of interest using a messenger RNA. Here's an example where we're microinjecting a capped messenger RNA that encodes 2 fluorescent proteins, GFP and DS-red, and we can generate fully developed larvae with both of these proteins. And we're now in the process of flipping out some of these fluorescent proteins to try to overexpress genes of interest. We're also have the ability to knock down genes with morpholinos and shRNAs. And here's an example of this using delivering shRNAs into one-cell zygotes, targeting brachyury, which is a known regulator of mouth formation. And so you can see that the animals with the shRNA fail to form a mouth and fail to gastrulate. And we also now have the ability to knock out genes with CRISPR-Cas9. And so we've been able to generate animals that are nearly completely mutant for the ammonium transporter in the F0 generation. And this will allow us to test fundamental questions about whether or not genes have a role, because making knockouts are kind of the gold standard for genetics. And so here are some of the uses of the tools that we've developed. I'm really interested, and the lab is really interested in trying to understand the molecular basis of the symbiosis establishment and maintenance. So are any of the genes that we've identified that are differentially expressed in Aiptasia, are they actually required for symbiosis formation? So we're systematically knocking out these several hundred genes in our larval platform to see if they have the ability to establish and maintain the symbiosis. Now, I told you at the start of the talk that corals are really hard to work with, which is true. but we care about coral bleaching. And so I wanted to start asking whether or not the things that we're learning in Aiptasia are also true on ecologically relevant coral. So I wanted to start to ask whether or not we could actually genetically manipulate corals themselves. And this started with a fantastic collaboration with Misha Metz and Marie Strader at UT Austin. And it's really continued with a good friend of mine, Lena Bay, at the Australian Institute of Marine Science. Now, unlike Aiptasia that spawn every week in the lab, corals generally spawn once a year in the middle of the night by the full moon, and the ones we were working with were in Australia. And so we had to pack up the lab and ship it to Australia and inject coral zygotes when they spawn during their natural rhythms. And we only had a few days to do this. And so we've been going back to Australia and now Hawaii over the past 5 or 6 years to try to genetically manipulate these animals with microinjection. And our strategy has been a really simple one, which is you make sgRNAs and Cas9 protein. I don't need to tell this group that. We in vitro complex these reagents. We inject them into one-cell zygotes, and then we also inject Cas9 only as a control for any microinjection induced phenotypes. And like I said, we've only had a few times a year to do this, but we've really made some significant progress in the ability to genetically manipulate corals. So our first year, we targeted 3 genes that we thought would lead to a phenotype. Spoiler alert, it didn't. But we were able to show that we were able to make high frequency of mutation, or high frequency of the animals that we injected had mutations. However, the animals that we injected were mosaic and had a low frequency of mutations in the larvae. So that precluded us from seeing a phenotype. We thought it was a big enough discovery that we went ahead and published it. Over the next several years, we decided to tweak the method. So we co-inject now with multiple guide RNAs that will target the same gene. We use a fluorescent injection indicator that allows us to successfully see who was injected. And now we also wanted to choose a target that would give a clear phenotype. And so we went back to Australia in 2018. And I thought that the HSF1, which is a known transcriptional regulator of that heat stress response that I was telling you about, kind of fits the perfect bill, because we know that HSF1 is a known regulator of the heat stress response. And so what I did in 2018 is we injected guide RNAs against HSF1 and 2 exons. And much to my happiness, with all these method improvements, during this 3-day spawning event, we got animals that were nearly completely mutant for HSF1. And we didn't stop there. We actually subjected them to heat stress. And so the experiment was injected day 1, grow up for 4 days, and then score survival during a heat stress trial. And this is the result, basically. The animals that were uninjected, the larvae, swam around nicely, but the animals that had mutations in HSF1 started exploding because they couldn't handle the heat. And we didn't just look at that one animal. We have triplicate experiments from each of the nights that the animals spawned due to their natural rhythm. So the animals that were uninjected or Cas9 only survived well during heat. So did the mutants at normal temperatures, but when we moved the mutants from normal temperatures to higher temperatures, they started to die. And so we're really excited by this, because it showed us that we were able to modify the genome of corals for the first time to make sufficient mutations to see a phenotype. And then we also showed that HSF1 is a key regulator of the heat stress response. And there's a typo on this slide. This was actually published last year in PNAS. Now, what have we done since then? Now, a lot of the work that we've been doing has been looking at larval expression, but we really care about corals and the adults that make these beautiful skeletons that are the reef-building organisms. So I wanted to ask whether or not I could expand these methods past the larval stage. And I started to ask whether or not, how do corals make a skeleton? Because I thought that would be a really obvious phenotype, say if a coral didn't have a skeleton. And previous research had identified a bicarbonate transporter that was expressed at the site of calcium formation, and the calcium carbonate skeleton needs bicarbonate in order for it to synthesize. And so I was interested in whether or not it was required for coral skeleton formation. So what we did is we took guide RNAs against this SLC gene, and we injected it into one-cell zygotes. We induced larval settlement and made baby corals that started to synthesize skeleton. And so after 2 days post-development, they start making skeleton that look like the spokes of a bike wheel. And at day 3, all the animals that could have made a skeleton would make a skeleton. And so we scored the animals for the ability to form a skeleton. And so the uninjected animals and the Cas9 injected animals make these beautiful skeletal spokes of a bike wheel, showing that they're able to synthesize biomineralization. And however, the animals that were mutant are not able to form that skeleton. So this shows that this bicarbonate transporter is a key regulator of skeleton formation. And this is really interesting because it turns out this bicarbonate transporter is only found in hard corals and not found in any soft corals. So the cool thing about this is it shows that maybe the co-option of this gene was a key step in the evolution of skeleton formation in corals. And we're in the process of publishing these results now. And we're also really excited because it shows that we can make genetic lines of corals, and we can look at phenotypes late in development. So we're now pushing forward to study genes involved in bleaching and symbiosis. And this is what the animals look like after a few more days. The controls have these beautiful castles of biomineralization, and the mutants no longer have that. So what's the future? So right now we're at Carnegie Embryology. We have the ability now to spawn corals in the lab. Actually, today we just got about 50 corals from the GBR in the Solomon Islands that we're going to try to get spawning at different times of the year, so we don't have to wait for the annual SPI cycle, that we can get spawning every 3 months in the lab for genetic manipulation, and we can actually grow out the animals that we've genetically manipulated. We're also trying to apply CRISPR into other species. So we spent 6 weeks in Hawaii this year trying to apply CRISPR-Cas9 to a new species of coral that's only found in Hawaii, to try to increase the number of species where genetic manipulation is possible. But our main lab focus is to try to take an integrative approach to understand cnidarian-dinoflagellate symbiosis. Now that we have all these genetic tools, we really want to understand what genes control the symbiosis and bleaching by studying both Aiptasia and then anchoring those discoveries in an ecologically relevant coral using CRISPR-Cas9. We have several positions in the lab for postdocs. So if you're interested in this type of work, I would love to talk to you. So feel free to email me. And I have to thank my lab and my postdoc advisor, John Pringle, and my friend and collaborator who we've been working on the electroporation protocol with, Chris Renicky. My many collaborations all over the world, and funding. And so I'm here to answer any questions if there's still time.
Wow, that was a very interesting talk, Dr. Cleves. Thank you so much. So we have a question at the Q&A session. So Ali is curious as to what sort of facilities do you use to cultivate Aiptasia in the lab? Could you tell us a bit more about it?
Yeah, so usually what we do is we use these little, I wish I had one. We have these little plastic shoe boxes that we fill with artificial seawater, and they'll grow great, right by a window. In fact, I realized when we were growing them next to a window that the animals would kind of climb toward the light. So they're an aquarium pest, and so they're really easy to grow in these shoe boxes. Here in the lab, we have made a flow-through system that will allow automatic water changes. And so all you have to do is feed. And so we have maybe 200 tanks, and we're getting a few more of Aiptasia that are growing, so we can keep lines growing and cut down on the husbandry. That's what we're doing in the lab. I should have had that in my talk, but I don't have it.
Thank you. We have another question from April. So she's asking, is there a method to target specifically the symbiotic genome with the CRISPR tool during the association with the coral host, or can you only target the Aiptasia or the Pora hosts? That's a
really great question, April. the dinoflagellates are really tricky. They're kind of like aliens. They have huge genomes. They don't have histones. They're tertiary symbionts themselves. People have tried to genetically engineer the algae, but they are not tractable so far. So all of our tools are trying to manipulate the host, but I do agree with you that we need methods to genetically manipulate the algae. And so we are trying to find some projects that might be advantageous, because that would open up a whole lot of really interesting biology. One thing we can do also is that we have different algal strains that in culture, and so we can infect different Aiptasia with different algal strains. They have huge effects on the host physiology, thermal tolerance, growth, and so we're trying to understand the genetic basis of that using natural variation, but genetic manipulation would be an awesome gold standard there.
Thank you. We have a hand raised by Mason. Go ahead, Mason.
Hi, Dr. Cleves. Thank you very much for your presentation. Earlier in the talk for your RNA-seq experiment, you had mentioned some of these transporters that are really important, and I noticed that one of them was a sterile transporter, and I was wondering if you wouldn't mind sharing a little bit more, if you knew much about the type of sterols that are being transported and the importance of those, and sort of those, in the importance of those in the ecology and of the association in particular.
It's interesting. Not much is known. We do know that these ammonium or these sterile transporters are actually pretty divergent in Aiptasia. Due to genome complexities, we don't actually know how many there are. But it seems like there's kind of been a radiation of these genes in these symbiotic lineage. We do think that the sterols are actually being transported to the host from the symbiont. We are interested in whether or not that transport is actually required for the symbiosis, and we don't know yet. There's some evidence that if you treat with pharmacological inhibitors of these proteins, that from human systems, that you can affect the symbiosis, but I think a direct genetic test would be really important to say what's going on. And so basically what we're doing is we're trying to systematically knock out various transporters to see what the effects of the symbiosis are. And I think this is a fundamental question: is nutrient transport actually required? And going back to April's question about the symbionts, we have all these strains of algae. Some form symbiosis, some don't. Some are taxa specific, and so it would be interesting to know what processes make some symbionts form symbiosis and others not. Maybe it's nutrition, maybe it's the degree of requirement of this nutritional exchange.
Okay, great. Thank you for your response.
We have another question from April. It's the same question even I have for you. Does the asymbiotic state of Aiptasia happen in nature, or is that the host state you have it in your lab?
April, that's a great question. One of the funny things about the Aiptasia is we don't really know very much what's happening to them in their own ecology. What's mostly known is what people are doing in the lab. We're trying right now with a collaboration with Corey Credit at Eckerd College in St. Petersburg to try to understand what Aiptasia do naturally. They're usually found on boat docks. We don't know exactly what's happening in the field. So are there aposymbiotic anemones in the field? I'm not sure. Now, there are other corals, particularly corals over where I am in Baltimore, that have a symbiotic and a aposymbiotic state that are found naturally. Those corals are often in environments where they can feed enough, and they have big enough polyps where they can feed and not need the nutritional requirement. That's an important piece of information for the Aiptasia. To keep the apos alive, we have to feed them. We feed both the symbiotic and the aposymbiotic anemones, but the apos presumably need to be fed or they'll die. That's a great question.
If anybody does not have a question, I have a small question for you, Dr. Cleves.
Okay.
So at what growth stage of the larvae do you do microinjection, and how do you replicate the environment, gray leaf barrier environment in your lab? And if that's the case, how long does the larvae stay alive after being injected with the microinjections?
Yeah. So for corals, we go to these natural events. So the corals spawn, that's usually induced by the full moon or the new moon for some species. They'll release the eggs and sperm into the water column, and then we collect them and fertilize them and then inject at the one cell stage. So we inject at the one cell stage to make sure our reagents are distributed uniformly across the animal. For corals, we can grow them up at least to their early juvenile stage. And so it's probably likely that we can grow up mutant animals and maybe even spawn them. In Aiptasia, we trick them, okay, in the coral spawning. So what we're doing in the lab with collaboration with a group in the UK, the Coral Spawning Lab, is we're trying to trick corals to think it's the right time of year by changing light, temperature, and the moon cycle. And so we think that we can take corals into the lab and grow them over the year and just trick them to think that instead of it's November, think it's March, and do that iteratively. And so that we should be able to get corals to spawn several times a year. And that's all playing with temperature and light and moon cycle. For Aiptasia, it's much easier. We feed them, we put them in the dark with a blue light, and they spawn a couple days later. So it's much more reproducible, and we can get them, and we also inject them at the one cell stage.
Okay, so it's just the way you trick them does work for them to spawn.
Yeah, basically, the blue light is really, really important. However, there's a few idiosyncrasies, a few things that we don't really understand about what causes spawning, both in corals and in Aiptasia. That'd be cool to study with new genetic tools.
So are you mainly working on Aiptasia in your lab, or is it like you are doing coral studies also?
So we have Aiptasia in the lab. We have coral in the lab. We just got corals today from 3 places in the Indo-Pacific. And then we also have an upside down jellyfish that has the same type of symbionts as corals in Aiptasia. So it's another system that we can hopefully get going in the lab.
So if anyone has any more questions from our attendees or panelists? We have one question from Brandon. Oh, sorry, we have a live question from April. When do you experiment like the RNA-seq on the cnidarian, or it's like the symbiote when you get that data back? How do you differentiate between the gene expression changes in the host gene versus the symbiote genes?
That's a good question. We do something very simple, which is just map to either genome. So you get the sequences back. They're both polyadenylated, and so we can map the RNA-seq reads either to the symbiont genome or to the host genome. There's not a whole lot of evidence for horizontal gene transfer between the symbiont and the host.
I think we do not have any more questions. So with that, I would like to thank you, Dr. Cleves, for such a wonderful presentation and enlightening us about Aiptasia and coral symbiosis.
Yeah, thanks. I'm really happy to be here.
Yep. Thank you once again.